So our laboratory has been studying the role of chromatin modification and chromatin composition in the regulation of gene expression. And of interest, we have been looking at the balance between the trithorax, the compass family of histone H3K4 methylases, and the Polycomb, the family of histone H3K27 methylases in regulation of gene expression. Recent studies of cancer genome have shown that mutations within the Polycomb and the compass family are associated with a large number of cancer. And Andrea Piunti here has discovered a role for a very first catalytic antagonist function of a gene within our genome that regulates Polycomb function.
So the Polycomb Repressive complex 2 is a methyltransferase that can methylate lysine 27 on histone H3 throughout the genome. So the activity of this complex is restrained by another modification that is DNA methylation. So when there is DNA methylation, there is no Polycomb activity. So these two different modifications in the genome are mutually exclusive. So as Dr. Shilatifard already mentioned, we discovered this new gene that is called CATACOMB, that stands for catalytic antagonist of Polycomb. This gene, interestingly enough, is DNA methylated, so is silenced normally in our genome. But when the DNA methylation level in the gene go very low, this gene can become expressed. It can bind to Polycomb repressive complex 2 and inhibit its activity. This is very important because unleashed activity of this complex, of the Polycomb repressive complex 2, may be spurious activity, may be like deleterious for the cells. So we believe that this gene CATACOMB works as a sensor to sense the level of DNA methylation in the cells that can be really low for different reasons. They can be physiological reasons, so during early development and placental formation, or pathological reasons, for example, during cancer. Interestingly enough, this gene is very poorly conserved throughout evolution, and the gene, this CATACOMB gene, is only present in eutherian mammals, which are placental mammals like dog, humans, chimpanzee, and so on and so forth. Into this protein, to CATACOMB, there is a stretch of amino acid, though, that is highly conserved among all these eutherian mammals. And this stretch of protein, a stretch of amino acid, strongly resembles the surrounding amino acid of a histone mutant, histone H3K27M, which is highly found in pediatric gliomas, that is like a very deadly tumor in children. So we believe that CATACOMB is actually the endogenous equivalent of this mutation and is regulated by DNA methylation.
Given that there is a catalytic antagonist function of Polycomb within our genome, this is very exciting news because there could be catalytic antagonists of trithorax and other histone modifying enzymes that their function could be harnessed for cancer therapy.